Our next speaker is Ben Ehrlich. Ben is going to be a junior at Torrey Pines High School. One of the requirements for this program is that the students have completed a year of chemistry, a year of biology, and they are at least 16. For the majority of high school students, that means they're coming in the summer after their junior year. Occasionally, though, those who have the lucky birthday placement during the year and were smart enough to take a science class their freshman year qualify a little bit earlier. So Ben is one of our younger participants as far as his grade. So he'll actually be going into his junior year next year at Torrey Pines. Right now he is looking at UCLA, Cal Poly SLO, Stanford. I think you had like 9 or 10 in your list. That's going to get narrowed down when you actually have to start writing them. Yeah. Guaranteed. Okay. Right now, it's not sure exactly what he wants to do. Obviously, he wants to be successful, and I think you will agree that this young man's going to be successful in whatever he does. Again, we have a scholar athlete with us. He just won the silver, your team? Yeah. His team won the silver medal in the JCC Maccabi Games, which is kind of a Olympics type of format, and his baseball team took the silver medal. So congratulations.
Hi, I'm Ben Ehrlich, and my mentor's name is Dr. Amy Rommel, and I worked in the Dr. Inder Verma Lab of Genetics, and I'm presenting correcting cancer in the brain. So the Lab of Genetics, as I mentioned earlier, is run by Dr. Inder Verma. He has been at the Salk Institute for a long time now, and what the lab is known for is they use gene therapy techniques to model disease and correct genetic mutations. They're able to do this by using stripped-down versions of different types of viruses. So cancer has been around for a very long time. The earliest evidence has been found among fossilized bone in ancient Egyptian mummies dating back to about 1600 BC. And although amazing technologies have been developed, no true cure has been developed. So my mentor, Amy, focuses on glioblastoma right here. You can see the tumor right there. It's the most lethal form of brain cancer. And it's extremely difficult to diagnose because there really are no symptoms for glioblastoma. And once it's found, it's usually in the later stages, and there's very low quality of life left for the patient. And as you can see by the minion illustration here, in purple is the tumor, the banana is its food, and the yellow minion is the blood vessel, and that's how the tumor relies on its nutrients. So recent therapy has failed tremendously. About a few years ago, there was a drug developed called Avastin, and it was thought to starve the tumor of its nutrients from blood vessels. It would actually kill the blood vessel, therefore the tumor would starve. So before Avastin, we can see the tumor here. It's smooth around the edge, somewhat contained right here. And then it's not shown on this slide, but it actually shrunk the tumor in a short amount of time. And then later on, as we can see right here, the tumor became more recurrent and invasive, and it actually grew. And as you can see here, in red, there's still blood vessels. And Avastin obviously didn't do its job if blood vessels are still there. So before Amy came to the lab, there was a researcher who did an experiment with Avastin. So he treated a tumor with Avastin. Here we have the tumor in green. We have the blood vessel marker, which is just normal blood vessel, and then the drug target, which is where Avastin works to basically kill off the blood vessels, and then the merge right here. And then after a period of time, we have the tumor here in green again, and oddly enough, the red overlaps completely with the green. The only explanation for that is the tumor created its own blood vessels, because if it glows green, that's the tumor, and then if it overlaps with the red and blood vessel. And plus, there is no drug target for Avastin to work, and Avastin only works for regular blood vessels. So that's the only explanation, is that the tumor grew its own blood vessels. So Amy asked the question, are these tumor blood vessels bad? Are they tumorigenic? Can they create a tumor if they're by itself? So what she did, she injected right here into a mouse brain with tumor blood vessels, and no tumor was developed. So to answer the question, no, these tumor blood vessels are not harmful. And since the tumor is harmful and it created something that isn't harmful, the tumor found a way to somehow cure itself in a way. So our approach for the project was not to kill cancer because it's our own cells. We're told from day one that cancer is bad and we have to kill it. But if you think about it, when you break your arm, you put a cast on, you treat it, reprogram it to its original state, and we wouldn't cut off our arm when that happens. That's how we basically treated our approach to curing cancer. So here we have minions again. They're crazy and sporadic. They're not really unified doing a function. So our goal is to reprogram it to do something useful in the body. So before I came to the lab, Amy did an RNA gene profiling right here. And this is basically determines the amount of RNA present in a sample. And RNA leads to protein. And the protein that she looked at was all the different proteins in the tumor and the tumor blood vessels. And this is important because protein makes up tissues and organs, and that's basically what the tumor is. So when I first arrived, she gave me a list of about 20 genes or so, and the JNK3 gene really stuck out to me because it's involved in differentiation of cells as well as cell growth, and that's what the tumor does, so that would make it potentially important to cancer. And it's also stress activated, which is, so the normal brain tissue undergoes a lot of stress when a tumor is present. So our hypothesis based on, this is relatively right here from the RNA profile, that JNK3 protein will be high in the tumor and low in the TDEX. We're just basically proving that the RNA profiling is in fact correct. So what we would do is we would see if JNK3 would increase in a tumor media, which is basically a tumor-like environment. So we'd like to see this trend right here, basically constant. And then in a cured blood vessel environment, we'd expect it to go down over a period of about 6 days. So to test this, we would use a technique called Western blot, which is basically used as a way to find the relative amounts of protein in a sample. Basically, after you're finished, you see bands, and based on its thickness, that's how much protein is there. So we ordered the antibody for JNK3, and nothing showed up. We're confused. It was all blurry, and no bands showed up. We did this not 1, not 2, not 3, but 4 times, and it all failed. So we weren't able to get sufficient data from that. So going to plan B, we were going to look at protein expression in slices of mouse brain. So the technique used for this is called immunofluorescence. Basically, you can see me here slicing mouse brains. Then we'd add primary antibody and then secondary antibody and then mount it onto the slide, and then we'd see where the protein is in relativity to the tumor. So antigens and antibodies played a huge role in immunofluorescence. So on the surface of the cells, there's strands of amino acids called antigens, and that's where the primary antibody binds to. So once we let it penetrate and bind, we add the secondary antibody, which binds to the primary, and that has a fluorescent tag attached to it, and that allows us to see the protein under the microscope. So what we found here is that JNK3 was localized around the edge of the tumor. So we can see this reddish pink is only around the green, and there's no overlap. And if we were to take out the green tumor, we can see there's a completely cut out space right here. And we can conclude from this that JNK3 is high in the normal brain tissue. It's high along the edge of the tumor because that's the only place where it's present. And JNK3 is not expressed in the tumor since there is no red overlapping with the green. So zoomed in even further, we have the nuclei of the regular tissue, the tumor in green, and then the JNK3 protein along this line, and then the merge. We can see it matches up perfectly along the line, thus confirming that JNK3 is around the edge. As well as down here, we have the nuclei again, the tumor, the JNK3 in red, and then the merge is fits like a puzzle how this goes right into here with this cut out right there. So to look at individual cells and where the protein was in relation to the tumor, we use a technique called tissue culturing, where we basically just grow and maintain tumor cells in a petri dish. And as I learned along the way, use of aseptic technique is crucial, or else you will ruin your cells and your experiment will fail. So what we found here is that JNK3 could potentially be some sort of tumor suppressor, because as we see in the tumor, this very faint red right here, whereas in the cured TDEC or tumor blood vessel, it's bright and there's a lot of it. So we came to the conclusion that the tumor somehow might be trying to push out JNK3, and yeah. So for final evidence, we found that so the bright red right here overlaps with the TDEC right there. We would have used a blood vessel marker, but our antibody was from rabbit, and the only blood vessel marker we had was from rabbit, so they couldn't coincide. And then the lighter red right here overlaps with the tumor. So in conclusion, our conclusion was the reverse of our hypothesis, unfortunately. So JNK3 was high in the TDECs, the tumor blood vessels, and low in the tumor. And JNK3 was mainly around both the tumor and the tumor blood vessel edges. So the possible source of error for our hypothesis being reversed. So DNA produces RNA, and the RNA goes through a process called translation, which in this case produces the JNK3 protein. But somewhere in the DNA, there could have been a mutation in JNK3, so that caused a mutation in the protein. So cells couldn't recognize JNK3 because it was mutated. It only recognizes it as its normal state. So it would order more JNK3 to be produced from the nucleus, and it would overcompensate, thus our results. So next steps would be, if JNK3 is increased in the tumor, we would have cured TDECs or tumor blood vessels would be produced. We'd do this by overexpressing JNK3 in the tumor to see if tumor blood vessels would appear. And since the RNA expression didn't match the protein expression, our hypothesis was reversed. We would sequence the JNK3 gene and look for any type of mutation in the gene. So I don't know what happened to that picture there, but outside of research was a lot of fun. It's supposed to be a picture of me pipetting at boot camp, where we learned basic lab skills, and without that we would have been thrown into a world-famous institute and looked like fools. So, and Amy also took me on Meet a Scientist / architectural talks, basically where we'd brief the tourists on what the Salk's main areas of research are and where they've been successful. And I actually got to speak about the LSSI program that I'm a part of and what my project was. Also, we met with a PI, Ed Calloway, and we learned, basically you don't get to meet with a PI every day because they're so busy. We got to just ask him questions about how he got into science and basically his areas of research and why he wanted to do what he does. Also, we went to Cancer Centers Council. We heard different scientists' approaches on how to cure cancer at Stanford Burnham. And then Amy is a part of the Society of Research Fellows, which is basically a group of postdocs and grad students that put on different events for the Salk. As well as the Salk picnic was a lot of fun. We played on, our interns made a volleyball team. And got a nice break from work, so that was a lot of fun. So first, I'd like to thank the Education Outreach Program, especially Donna and Ellen. You guys were awesome. As well as the Laboratory of Genetics, Dr. Amy Rommel and Dr. Inder Verma. Your guidance and knowledge was super helpful. I don't know what I'd do without you guys. As well as the entire lab, helping me just find little things here and there. The LSSI professors, Craig Milgram and Allison Shear. Without them, as I mentioned earlier, I would have looked like a fool without knowing the basic lab skills. All the Salk scholars, who I got to know really well over the summer. Also, Mom, Dad, and Kara, and the rest of my family. Thank you for listening.